to thank the organizers for providing an opportunity to talk about a big project that we are running for several years in a group. So I'm taking you to networks, as we discussed before, just to a different scale of that. So what is the objective of constructing a signaling network? So first of all, the generation of an organized resource containing formalized summary of biological results from many research groups. Such resources provide platform for sharing information and discussing processes depicted on signaling networks. Of course, these signaling networks are not only cartoons, and they are basis for analytical tools and for omics data integration, and also for modeling. So those signaling networks that can be well found in many databases, actually, like Reactome, KEGG, or alternative resource that I'm going to represent to you, they can be involved in a typical systems biology project when some observation is done in a lab or some scientific question is present. And then signaling networks in the formalized way can help to analyze the data, to interpret the result, to perform modeling, and suggest particular mutants or particular interventions, and come back to the lab explaining the observation and raising a hypothesis and explaining a molecular mechanism that, of course, has to be validated in the lab, and this circle will continue. So, since I'm coming from Institute Curie, which is actually a cancer institute, our interest is signaling involved in this disease. Cancer is a very complicated disease, and practically everything is interfered there. Given the fact that all signaling in a cell is talking to each other, it's very important to ensemble and represent cell signaling together. And then our aim is to depict complexity of cancer. For that, we initiated Atlas of Cancer Signaling Network. We have used hallmarks of cancer, those processes that are implicated in majority of cancers. Behind each of these hallmarks, there are particular mechanisms that we represented as maps, and together with maps, in analogy to geographical maps, contain atlas of cancer signaling networks. So the map is huge, therefore we decided to build it in a hierarchical structure. So there is a global map, which is seamless tissue of the signaling, but it divided to maps of biological processes, which further divided to modular maps, and each module actually represents a canonical pathways as we used to see them in other databases. So those maps are manually constructed using latest literature and other resources. So we use the systems biology graphical notation syntax to represent the processes, and we do it in Cell Designer tool, which is very familiar for biologists because the major specialists that are doing those maps are actually biological students. So we represented in the form of biochemical reactions with a depth description of all details, which then translated to SBML format, making those maps interactive and also amenable for computational analysis. So the data model that we use in this resource is process descriptive diagram, showing step-by-step all biochemical reactions, regulations, and post-translational modifications. This data model allows also to represent compartments of the cell. This is a resource. It's an open source. You can visit the website. Of course, it's an ongoing process as any other signaling resource because knowledge is coming up every day. So you can learn about statistics a bit. So far, there are 5 big maps composed of 52 functional modules. They're all interactive. They're all interconnected. And the map can be observed as a whole, or each individual submap or submodule can be visualized as a separate map with the unique separate layout adopted for better visualization. Well, of course, when one constructs such a huge map, it's an effort as it is, but the next effort is to navigate through the map. Like in geographical maps, we decided to profit from Google Map Engine and explore our maps as Google Maps. We have created Navicell tool, which uses Google Map Engine. It's associated with a blog, and it contains also a toolbox for data integration and analysis. In terms of navigation, the tool allows to zoom in on the maps, to visualize in pop-up window external information on each entity. And we suggest several ways of navigating through these big maps. Horizontal map navigation is shuttling between global maps to modular maps, like in this example, and following the entity of interest, which will be marked by markers, as in Google, observing also the annotation of the entity, with many hyperlinks to other resources where the extended information on the entity of interest can be retrieved. Vertical map navigation is simply zooming in on the area of interest, and we implemented here a semantic zooming principle where at each zoom level only essential level of information is shown, starting from a top-level view where only structure of maps are shown, up to a very detailed one where all regulations are shown. An additional way to explore for the maps is highlighting the neighborhood of the entity of interest. For example, here you see B53. So by applying this search neighborhood function, one can see how the signal propagates from the entity of interest through the map. So that's all about the navigation that's provided by Navicell tool, which supports ACSN resource. Of course, these maps are not only cartoons, as we said, we are using them for integrating and interpreting data. So the toolbox, which supported by Navicell, allows to visualize continuous and discrete data. This is the first example of visualizing frequently mutated oncogenes and tumor suppressor genes. So the frequency of mutations are shown here by colors. And here, at the very top-level view of the maps, we simply see a distribution of mutated genes across different functional processes, which demonstrates, for example, that tumor suppressor genes are mostly appearing in cell cycle and DNA repair. And actually, oncogenes are almost not seen there, which is biologically very relevant because normally these processes are protecting against the development of cancer. And of course, these genes involved in these processes would be modified by cancer cells to break through and to develop a cancer. Of course, these maps can be zoomed in, so when data is projected on the maps, the zooming functions are there, and then each node with a corresponding box or the circle will be visualized at every zoom level of the map. Here is a different technique of visualizing data in the context of big maps. We call it map staining. So instead of visualizing individual values corresponding to each entity, here we visualize enrichment of functional modules. So here we used a transcriptome data from TCGA database for 4 types of breast cancer, and we calculated a module activity based on the expression data according to the involvement of different entities into functional modules of the maps. And then the value of this enrichment is projected on the map, actually giving us an impression of activation or inhibition of different functional modules, but not the individual entities. This is very informative because we immediately can grasp the differences in the involvement of different processes in different diseases. As I mentioned, we depict processes in a very detailed level and we also depict the post-translational modifications. The most frequent one is phosphorylation, of course, but of course we would represent other types. Here, what is demonstrated is phosphoproteome data projected on the corresponding entities with the corresponding residues modified. And the color represents the frequency of the particular phosphorylation across the studied cohort. The last slide about visualization is that Navicell allows to combine several types of data and visualize them together. As you know, there are many different types of omics, and the question always is, what can we learn together from this data? And then the first impression of this data can be done using Navicell. So here in this example, the expression data is visualized as map staining. It's an enrichment of functional models as a background. The copy number variations are shown as heat maps, boxes, and then mutations are shown as triangle glyphs. And then, so these molecular portraits, we call these pictures molecular portraits, actually give us an impression of what is affected. Then one can go and dive into the area of interest where something is most affected, most interesting to the researcher, and then search for the processes that are most involved in a particular disease or in particular treatment, depending from what source the data is taken. So that's it about visualization. Now I will give two small examples on how these maps can be used. So in first example, we address the question of creating transgene mice of invasive colon cancer. So according to the current status of the field, despite the fact that colon cancer is very much studied, there is no good mice model with early metastasis. And then to help to develop such a map, we constructed such a mice, we constructed a map of processes involved in induction of metastasis. And when we studied key players and performed a path analysis of the map to understand what genes we have to interfere with to achieve a desired phenotype, metastasis. We used that model for mathematical modeling, and we interfered with each gene or each pair of genes on this network, in silico, of course. And applying these logical rules, we actually arrived to the conclusion that in order to achieve a metastatic phenotype, one has to interfere with a notch or its cleaved product NICD together with P53. When P53 is downregulated and NICD is upregulated, these are the only conditions that will permit induction of the process responsible for metastasis. So, and then we suggested a mechanistic explanation of that, and this hypothesis has been validated in mice that shows very early metastasis in a distant organ, which originates, of course, from colon. In another example, I will start with explaining the principle of synthetic lethality. Synthetic lethality is a phenomenon when interference with one gene does not affect cell phenotype, the same for the second gene, but when two genes are interfered together, then it causes cell lethality. This is a synthetic lethality paradigm, which is used in treating a particular type of breast cancers. So those patients that carry mutation in BRCA gene are responsive to inhibition of PARP molecule, creating a synthetic lethality. However, many of those patients develop resistance to treatment. The question is why? After constructing big signaling networks, one can understand that, that there are many redundant pathways on the networks, ensuring robustness of signaling in a cell. And then it easily explains why the pair-based synthetic lethality might not work, because in reality, we have to switch our understanding from synthetic lethal pairs towards synthetic lethal sets in order to interfere with the process of interest and to achieve a particular phenotype. In our case, we would like to achieve interference with proliferation and stop development of cancer cells. So in order to find what would be the best intervention gene set instead of pair, we developed a tool called Oksana that calculates the minimal intervention sets using a structure of the networks, allowing to interfere with the node of interest or combination of nodes of interest. So using this tool, we suggested how to restore sensitivity for those patients, in those patients that are resistant to PARP treatment. For that, we used DNA repair network because both BRCA and PARP are playing a role in DNA repair pathways. So this is a DNA repair network from our resource. Here you see a backbone structure of this network. So here you see the node of damaged DNA, and then these paths are actually different ways of repairing DNA damage up to the intact DNA. So here by these crosses are indicated places where PARP is involved. Even by inhibiting PARP in the context of BRCA mutation, there are still enough paths to repair DNA, and this most probably explains resistance to PARP inhibition in patients. So how to suggest better treatment, more complex treatment? So we calculated all possible intervention sets with these DNA repair paths, and we retrieved all those sets that include PARP. So, of course, one ends up with several tenths of such an intervention sets. And to suggest a treatment, we need to come up with one or two suggestions. How to prioritize those intervention sets? For that, we use the internal vulnerabilities of the patients. How we can learn it? Of course, from omics data. This example is based on cell lines retrieved from these patients, which are BRCA mutated, but resistant for PARP treatment. After integrating these omics data into the networks, we could prioritize intervention sets. Here is an example. This is a triplet suggested as optimal hitting set. In this particular patient, XRCC4 is already inactivated due to the mutation. And therefore, the remaining gene to interfere with is MSH3, together with a PARP inhibitor. So this is a suggestion of personalized treatment for that particular patient. The other possibility is this triplet containing RAD50. This is another personalized treatment that can be suggested to that particular patient. So using this example, I am introducing to you the pipeline which we are developing now in the institute together with a hospital, where we use Atlas of Cancer Signaling Network, our resource, and we integrate data from patients. There's a lot of cohorts available in the institute. And we develop, first of all, we look at the molecular portraits, as I introduced to you several slides ago, to understand what are the most involved processes on which we will be looking for intervention sets. We perform structural analysis and we suggest intervention sets. We prioritize them, as I just explained in the previous slide. And then these intervention sets, of course, have to be validated in preclinical models. If successful, they can be taken into the clinical trials. So we developed with this pipeline to be a basis for personalized medicine in the institute. An additional added value of such a procedure that we can learn modes of synthetic interaction mechanisms in different cancers. And then hopefully we will be able to come up with a drug sensitivity predictor if we learn sufficient number of patients and different cohorts from different cancers. The same paradigm can be actually used not only based on Atlas of Cancer Signal Network, but any other networks, and can be applied not only for cancer, but also for other diseases or analyzing other data sets from experiments in the labs. At this point, I would like to thank our multiple collaborations, and these are people involved in ACSN and Navicell team, and I will take your questions. Thank you.
All right, do we have any questions? No?
Everything was clear.
All right, if not, let's thank the speaker again.